الشيطان الرجيم بسم الله الرحمن الرحيم انطلقوا إلى ما كنتم به تكذبون انطلقوا إلى ظل ذي ثلاث شعب لا ظليل ولا يغني من اللهب إنها ترمي بشرر كالقصر كأنه جمالة صفر ويل يومئذ للمكذبين هذا يوم لا ينطقون ولا يومئذ لهم فيعتذرون ويل يومئذ للمكذبين صدق الله العلي العظيم. Lecture number 48 for the 9th class with a term lysosome. Lysosome. I was not expecting that this term will be included in 9th class because this term is being explained in senior classes. We explain normally. But if you have studied Golgi complex, this term is very easy. First of all, I will go to Golgi complex and I will tell you Golgi complex. No, later on I will tell you because lysosome. What are the lysosome? They are just Golgi vesicle. So I spoke one term that lysosomes are just Golgi vesicles. So don't worry. What are the Golgi vesicles? They were formed from the Golgi apparatus. Those were the products which were packed in a vesicle, and those vesicles are called Golgi vesicles. And which products were formed in Golgi vesicle? Enzymes. Now I am telling you very important information. If you are attentive, please. Enzymes, if enzymes in Golgi vesicle are enzymes which are used in digestion, mean they are digestive enzymes, mean they are changing large particles, lipid, fats, carbohydrates into small molecules, mean they are changing lipid to fatty acid, glycerol, they are changing protein to amino acid, they are changing starch to glucose. It means that those Golgi vesicles are called lysosome. It means there is no difference between a Golgi vesicle and lysosome. They are basically lysosomes if they consist of digestive enzymes. Digestive enzymes I will name once again: amylase, present in oral cavity, you will study in digestive system, which change the starch to glucose. In the stomach, enzyme is pepsin, which changes protein to peptone. Peptone I will teach you in digestive system. Amino Peptidase, another enzyme you will study in the 9th class digestive system chapter. Amino Peptidase, which change the peptone to amino acid. Similarly, trypsin change the trypsin change the protein to the peptone, and lipase change the lipid to fatty acid and glycerol. So digestive enzymes are the lysosome. Why the lysosomes are present? These organelle is new organelle, just discovered now, because this is a one membrane organelle, not a double membrane organelle. Name the double membrane organelle in your cell. Nuclear membrane, double membrane, of course. Nuclear membrane is also called envelope, nuclear envelope. Name any other double membrane. Mitochondria, double membrane. What is the name of inner membrane? Cristae. Name any other double membrane structure. In the cell, chloroplast. Name the inner membrane, thylakoid membrane. So this is a single membrane. Lysosome is a single membrane. So this is used. How it will be used? He is not telling the figure. I have to draw. I have to make the figure of this one. I just checking one thing. Just hold on. No, he is not going to. He is not going to explain. So I have to draw this figure here. Right here I will draw. Look here. This is a Golgi complex. Golgi apparatus, right? Golgi apparatus. Golgi apparatus, you know, is making the product. Product is enzyme, and enzymes are packed in a vesicle. And which vesicle? Which vesicle? Golgi vesicle. Clear? Okay, these are the products. These are the products. If these products are digestive enzyme, if these products which is inside the Golgi vesicle, here I am naming. If these products are Golgi vesicle, if these products are digestive enzymes, so this Golgi vesicle is called as lysosome. You are getting the definition of the lysosome, or I need to explain? I will explain. I am explaining all terms Don't worry, cell wall is not finished, cell membrane is not finished, primary cell wall composed of cellulose not finished, I will revise, and secondary cell wall composed of lignin not finished, sclerenchyma dead cell not finished, collenchyma living cell not finished, the bacterial cell wall composed of peptidoglycan not finished, fungal cell wall composed of chitin not finished. I will explain again. Bilipid layer in the cell membrane not finished, bilipid layer and the protein discontinuous layer not finished, bilipid layer allow non-polar molecules to enter into the cell not finished, cell membrane is a semi-permeable membrane not finished. I will explain again and again these things. The model explain the structure of cell membrane, that is a fluid mosaic model, and the mitochondria inner membrane cristae not finished, F1 particles not finished, ribosomal unit, large ribosomal unit, small ribosomal units not finished. I will explain again and again in the physical classes also. In the physical classes also I will explain also further, because you know when you will revise these things again and again, the memorization become easier. Dear students, that is the point. Golgi complex I will explain again, the membrane of Golgi complex I will explain again, that is the cisternae of course, and the transport vesicle getting the enzymes from the rough endoplasmic reticulum. I will explain these things again, and what will happen, the enzymes which are present in the vesicle, that is called as the Golgi vesicle, and Golgi vesicle consists of digestive enzyme that is called lysosome. That is the point. Nucleolus I will explain again, the transfer RNA, messenger RNA, and the ribosomal RNA, and the chromosome, of course, the hero which is present inside the nucleus, which is composed of a significant material, hereditary material, which is the DNA, and a protein which is a stone, which is positively charged, and the liquid inside the nucleus, nucleoplasm, and the liquid inside the mitochondria, mitochondrial matrix, and the liquid which is present inside the chloroplast. Do you remember? Stroma. Yes, the stroma, stroma. I am talking about the lysosome now. I am talking about the lysosome. So what is happening here in this case, the Golgi complex will produce enzyme, will pack in a packet, and the packet is called as Golgi vesicle. That packet is called as Golgi vesicle, and these are the enzymes inside Golgi vesicle. If the digestive enzymes present, this packet is called as lysosome. So Golgi vesicles become lysosomes when it consists of digestive enzyme. The point, very important point, I did not tell yet, I am telling you now. Are you alert now? I am telling you now. Look here, I will rub all these labeling, because then I will be able to explain, because I want to introduce another hero. One is a food vacuole. Do you know I told in the animal vacuole cell, I told you animal vacuoles are of two types. One is a food vacuole, one is a contractile vacuole. Do you remember? Do you remember the two vacuoles I named in the animal cell? In the plant cell, I told vacuole is very large, that is present at the center, and that creates the pressure called turgor pressure. I did not tell turgor pressure in the animal cell. I divided the vacuoles into two types, food vacuoles, contractile vacuoles in the animal cell. I told one is a food vacuole, and I told this phenomena also, food vacuole consists of the food. I also told food vacuole is not present in the humans. It is present in these less developed cells, less developed organisms. Less developed organism it is present. Less developed organisms it is present. And these molecules which are present in this vacuole, which is the food vacuole, you know, which food you eat? You eat protein, you eat fat, you eat carbohydrate. Protein, fat, carbohydrate you eat. These three molecules you eat. Protein you eat, fat you eat, carbohydrate you eat. This protein molecule, fat molecule, carbohydrate molecule you are eating. This protein will change into which product I told you? Amino acid. Yes, you are right. Fat will change into fatty acid and glycerol. What I am doing? I am changing large molecule into small molecule by help of enzymes. Large molecule into small molecule, yani mean catabolism. So why it is called digestion? Because when protein, fat and the lipid goes into small molecule, that is called digestion. That is the digestion, and that's why the nature has made digestive system. So why sir food vacuole? Because digestive system is made, digestive system made by Allah. So digestive system is breaking lipid into fatty acid glycerol, digestive system is breaking protein into amino acid, digestive system is breaking carbohydrate to glucose. So why sir there is a use of food vacuole? Even digestive system is present in the human body. Students, I am not talking about the human body. In the human body food is digested in digestive system. I am talking about the small organisms like amoeba, like paramecium, which are very small, one-celled. They have no digestive system, they have no stomach, they have no esophagus, they have no intestine, they have no digestive system. So they will digest their food in this small structure, food vacuole. You are getting me now. You are getting me now, students. I am just rubbing some terms and then, okay, two vacuoles. One is a lysosome. What is a lysosome? This is a Golgi vesicle which was produced by the Golgi apparatus produced a product, and product was packed in a vesicle, that is a Golgi vesicle. And Golgi vesicle, if digestive enzymes, these black colors are, mean these enzymes are used for digestion, so Golgi vesicle become lysosome. Now what will happen next? What will happen next? They both will combine. They both will fuse. They both will fuse. I need your attention, students, because I am talking about the lysosome. Technically, this lysosome is called primary lysosome. If you will understand this phenomena, I don't need to teach this topic in much detail in first year, because you are getting the data of the first year now. Because when I will deliver the lecture of this topic in the first year, you will feel very easy, because these informations, almost same informations, are in the first year. Primary lysosome. They combine with the food vacuole. They will combine with the food vacuole. Why they are combining with the food vacuole? Because they consist of enzymes, and these walls between them will break down. These walls between them will break down. These walls will break down, and these walls will break down, and food vacuoles will, enzymes will move from the lysosome. Which lysosome I speak? Primary lysosome. To the food vacuole. To the food vacuole. So conversation will not finish yet, because you know I am just using the two terms now. Before fusion that was primary lysosome, after fusion it was the secondary lysosome. So you will name, you don't need to name even the 9th class, because these are the terms of the first year or second year class. You can say simple lysosome if you want to say, no issue. But just for knowledge I am telling, this is not much difficult, yaar. If the lysosome combined with food vacuole, it is secondary lysosome. Before that it is a primary lysosome. Okay, the enzymes goes into food vacuole for digestion, because they fused with each other, and these enzymes which come from lysosome to the food vacuole, this is a food vacuole, this is a food vacuole. So food will be digested here, and this digested food after digestion will go out to the cytoplasm to provide the food to the cell. And I will explain now. This is a secondary lysosome, mean that food vacuole and secondary lysosomes, they have combined. Enzymes come from lysosome to food vacuole. This is a food vacuole, this is a food vacuole, this is a lysosome, and food is digested here, mean large molecule break into small molecules, mean protein changes to amino acid, the fat changes to fatty acid and glycerol, starch changes to glucose, and that digested food enter into the, will diffuse. So food vacuole is so important. This is the food vacuole. After digestion, mean after getting the enzymes from the lysosome, it will diffuse. I want to define later on, but I will define little a bit now. Movement of the molecule from the high concentration to the low concentration is diffusion. I was just not defining the diffusion. Reason is that I want to differentiate between three types of the processes in the coming session. One is the diffusion, one is a facilitated diffusion, one is a osmosis. But simply you will understand that the movement of the molecules from the high concentration to the low concentration, that is the diffusion. That is the diffusion. So diffusion move from the high concentration to the low concentration. Molecules move from the high concentration to the low concentration, and these molecules which move from the high concentration to the low concentration, and they will move into the, these molecules will move into the, what will happen, they will go into the cytoplasm and digested food. You know why digested food is needed? For energy production. Because when starch changes to glucose, the glucose, you know, that is digested food. Why it is digested? Because that will take part directly in respiration. What is the respiration reaction you studied in this chapter? Glucose react with oxygen. You will understand this thing. Which thing is required to produce energy? Glucose. Who will produce glucose? When starch changes to glucose. So starch changes to glucose is called as digestion. So why digestion is necessary? To change large molecule starch to small molecule glucose. So why glucose you need to form? Because glucose can take part in respiration. Why glucose you want to participate in respiration? Because that will produce energy. And why energy? Because energy will be produced, we will move. So this digested food, being glucose, will go into the cell cytoplasm, and then it will go to the mitochondria, you know, and that will participate in respiration. So that by this way, food vacuole, lysosomes combine and they become one structure. Food digested, food comes out, food goes to mitochondria, food participated in respiration reaction, glucose plus oxygen, carbon dioxide and water, energy is released. That energy is used for our body activities. This was lecture number 48, students, and lysosomes and Golgi complex, and these were very important structures because they were forming enzymes, and enzymes were reacting with the food, with the protein, and enzymes were changing protein into amino acid by pepsin, fats into fatty acid glycerol by lipase, and the trypsin or pepsin changing protein into peptones. And amino peptidase, another enzyme which is changing into peptone into amino acid. These are all enzymes you will study in digestive system, not written in this chapter, but here you got the concept that food vacuole need enzymes from the lysosome. Thank you very much. This was lecture number 48.